I just want to say welcome to Texas. I'm so happy to have you all in my home state. Hey, y'all, I'm Brendan Hayes. I'm the Director of Education for Innovative Therapies, and I think we're going to be in for a real treat today. So I'm going to just get started and introduce Dr. Robert Sidonio with Emory University. He's going to do our, it's about a 40-minute presentation. Immediately following, we're going to have a panel discussion. Thank you very much, Dr. Sidonio.
All right, good morning. So that's my fault for using the wrong aspect on the slide. So you're not going to get everything. So you're just going to have to deal with it. Sorry. All right, so my task is really to cover some advancements. And, obviously we're talking about all bleeding disorders. So I don't want to make this a 45-minute talk about just hemophilia because, it's important that we talk about advancements in other disorders as well. And, and we're going to verbally discuss gene therapy at the end, just for the sake of time. It's really hard to cover everything that's going on in the bleeding disorder world. So let's go ahead and get started. Hopefully. I don't know if it's not advancing or, oops. You want to talk about anything while we're waiting for the slides? Is there a particular topic? So it's been pretty good weather here. A little drizzle last night. So, all right, let's get started here. All right, so we'll start out with the disclosures. I'll leave that up there for a minute. All right, so we'll talk a little bit about new drugs in the pipeline, and I didn't want to do an extensive review. Just really want to touch on these topics, and obviously there's plenty of time at the end of this for questions. And we can further explore some of the nuances of this. So one of the things, I feel like, and I wanted to start this off with Glanzmann's thrombocythemia. With so many good therapies in hemophilia right now, like Shannon and I always talk about, this is the most difficult disorder to manage in our clinic right now. One, mainly because we don't have really good therapies for this. We've basically had the same therapies for decades. And as you remember, this is a very rare bleeding disorder. It's autosomal recessive. The defect is right there, specifically in this receptor, the glycoprotein 2B3A. And the diagnosis is fairly straightforward if you have platelet aggregation studies. It can also be diagnosed by flow cytometry. You have to be careful. It doesn't pick up all of the defects on flow cytometry, but we're often able to identify children. I think we identified a child at 6 months of age recently. So you can do it, and you don't have to wait for the aggregation studies for them to get old enough for it. But this critical interaction here where fibrinogen is bound leads to a lot of bleeding. And the manifestations are pretty significant. GI bleeding, nosebleeds, the worst nosebleeds you would ever see in a patient are in Glanzmann's, probably the most scary nosebleeds I've ever seen when I was a fellow in Pittsburgh. Oral bleeding, heavy menstrual bleeding, muscle bleeds, and a lot of skin bleeding. And so just causes a ton of bleeding. There's a pretty big variety in bleeding. We have some patients that have very little bleeding and some patients that bleed just about every day of their lives. The mainstay of treatment has been platelet transfusions. If your platelets don't work, you can get somebody else's. And then, recombinant 7A, and because there's a new product on the market where you can distinguish them, this is Novo7 or Epticog Alpha. And then we always talk about boatloads and boatloads of Amicar and Lysteta. I mean, sometimes you just say you want people to take a bath in Amicar. It's like the only way to get some of the bleeding to stop. And so I'm going to go off label real quick here at this point, because this isn't a space where everything is just about off label, because on demand, Epticog Alpha is not adequate. Anybody thinks that, it really hasn't taken care of a patient with this. So one of the things that we looked at our center was this concept, again, this is off label, of using 7A as prophylaxis. The dose, we just pick the dose that's on label for an off-label indication. Oftentimes, these patients are on it so frequently, insurance companies don't even notice they get it 2 to 3 times a week, because when they have a bleed, they get about 20 doses, and they often get multiple bleeds a month. And Dr. Batsuli led this effort here at Emory, but you can see these are patients with Glanzmann's thrombocythemia, estimated ABRs from a chart review. And so some patients, particularly this one, had a pretty dramatic improvement in the bleeding phenotype. And most of the patients felt like they had a little bit more control of their bleeding, even if they felt like their total bleeds may not have gone down significantly. And so this is something that it's not just being done at our center. There's a number of centers that have attempted this, and something that should be hopefully under consideration. And based on that, we have an early study that we're looking at, and we're going to use epticog beta or 7-fact, and we're going to try to see if we can use this prophylactically. This is one of those sites where you don't need a lot of one of these studies. You don't need a lot of patients. This is a super rare disorder. We're going to compare it to historical bleeds, and we hopefully to start in 2023. So really would like to look at whether prophylactic infusions of epticog beta, potentially on demand as well, may help with this disorder. And really this is a good, this is like a nice thing to get for patients, hopefully to be able to improve their lives, make their lives a little bit less chaotic. Even more excitingly, there's a product made by a company called Hemab. And this company makes a lot of monoclonal antibodies, very similar to what emicizumab is. And this product, I keep telling them they got to take the HMB, because every time I see it, I think it's heavy menstrual bleeding. But it has nothing to do with heavy menstrual bleeding. But HMB1 binds and accumulates factor 7A. So if you look right here, this is a subcutaneous injection, very much like emicizumab. And what it does is it binds to factor 7, and then it brings it to the site. It binds specifically to this receptor that's found mostly in megakaryocytes and platelets, so it should be very specific. So it's bringing your own factor 7 to the site of damage and allowing it to hopefully have improved hemostasis. They showed some data at ISTH, early data. It looks very promising. This will be a subcutaneous drug. We don't know the exact frequency of it, but it'll probably be fairly similar to what you see with emicizumab. One of the challenges I think we'll have to see is how avidly does it bind here? Is it going to allow factor 7 infusions to be accepted, much like you see with emicizumab, which there's they've designed it where it won't bind really tightly. So early discussions, but it's exciting that this is actually going to be considered hopefully sometime early next year. Right now, it's past the discovery preclinical phase and entering the phase 1. So we'll see what happens, and potentially they're looking at whether this could be useful for other disorders like Bernard-Soulier syndrome as well, which has no other therapies as well. So what about trials in von Willebrand's? I specifically started with other disorders outside of hemophilia. One of the interesting topics, and we'll get into this a little bit more when we talk about hemophilia, there's a drug called, used to be called BIV001, now it's called efanesoctocog alpha. Getting better at saying it every time I do these presentations. But what it does is it limits your interaction with your own, it's a factor VIII product that limits the interaction with your own VWF. And by uncoupling that interaction, it allows it to extend the half-life much longer than any other extended half-life product. They took a portion of VWF to bind to it, so it won't bind to VWF. They added these biodegradable products called X10 linkers. They're these just short amino acids. And it's sort of it's almost like an improvement off of a pegylation, a little bit easier to degrade, but also provides the same properties by making it larger and difficult to eliminate. When there's activation via thrombin, you end up with aloctate. And the data on this looks very interesting. The most, and we'll talk a little bit about this, but patients were able to sustain a half-life of about 47 hours, which is significantly better than the best extended half-life product, which is about 17, 18 hours, depending on if it's median or mean. And then you're able to achieve quite good levels. You actually have near normal levels for 4 days and a trough level of 15%. So you spend most of your week above the normal range, and there doesn't seem to be any accumulation, and the clearance seems to be the same regardless of age. So the interesting thing is some of the original studies in von Willebrand's were actually in fractionated plasma, and they were actually giving mostly factor VIII to these patients early on, mostly because they didn't know about VWF back then. And so, we kind of go in almost full circle back to the 1950s and 60s. Is there a role for factor VIII in severe von Willebrand's? And so Sanofi is going to take a look at this. They're going to look at the pharmacokinetics and safety of this, and they're going to pick disorders that specifically have very low factor VIII. So typically type 2N, If you're a homozygous type 2N, you have very low factor VIII levels. And so they're not going to look at efficacy quite yet, but this is something they're exploring. I don't know if this is going to be useful, but it's certainly worth considering. And so there'll be 9 adults that should be type 3 and type 2N von Willebrand. So this is something that's currently underway, something to think about. Additionally, there's been this concept as well about emicizumab and whether there's any role for non-factor therapies in von Willebrands. Currently we only have intravenous products for von Willebrands. And the idea of this comes again from the concept that ultimately you just want to form thrombin, you want to form a nice fibrin clot. And sometimes the way to do that is actually to get the factor VIII level up. If you look at the old von Willebrand guidelines, the dosing is actually all based on factor VIII dosing. And you can definitely predict more bleeding as the factor VIII level goes down. But this is what a typical thrombin generation curve looks like. You infuse, you get a nice peak. It looks very left shifted. And then there's this nice area under the curve. And when they've looked at this in patients, so this is ones with no treatment here with a severe von Willebrands, as they give more doses of emicizumab throughout the study, they're able to replicate normal thrombin generation. Of course, it doesn't affect VWF in any way, and it's not going to protect you from all bleeds, but it may be enough to mitigate some of the bleeding and make things a little bit better. And there have actually been a number of cases reported. So this case series had 2 cases reported here. And you can see here, the patients here, these are all severe von Willebrand patients, and 3 of them actually have inhibitors. They tried to quantify one of them here, and all of these have cross reaction with factor VIII. And so these patients were placed on emicizumab and had pretty significant bleeding, but continue to have some bleeding episodes. So it's not perfect, but obviously it is encouraging that they have an option, because these patients had no options, particularly the ones with inhibitors. So another concept that's being explored as a drug class called aptamers. And a lot of these drugs have been created in other disorders. So I've been using these short-stranded RNA and DNA molecules, and they've been using these as chemotherapeutic agents. They've been using this in HIV as well. And the idea is that these are going to be fairly low cost. They bind very easily. They're very specific. And the cost of production is low. And so this product here called BT200, you can see the binding here in this ribbon structure to VWF. It specifically binds to one part of the domain that overlaps with the clearance receptor. And what we at least think, based on some studies that have been done by Dr. James O'Donnell in Ireland, is that it decreases the clearance from circulation, because it doesn't seem to affect production at all. They've actually looked at propeptide levels, and it doesn't seem to affect that at all. The great thing about this is it's a subcutaneous injection. These things are typically very unlikely to have some immunogenic reaction as well. And so it looks very exciting as a possibility. Probably the most exciting thing is that we've been trying to work with this company, and one of their big trials actually is potentially going to be in women and girls with hemophilia and women with von Willebrands. And so we're trying to get some of the companies to consider this population as the first population to study the drug in their pivotal trial. And that's not been done previously. So we're excited to hopefully get that going. A little bit of data on this. This was presented at ISTH last year, and you can see the subcutaneous injections here. And these patients were normal patients, but you can see how high you can get the VWF levels here. And obviously this is not what you want to do in a normal person to get the levels over 200, 300. But we know that it can elevate the levels significantly. It didn't affect the propeptide levels. So it really just seems to keep your factor around in your body a little bit longer. So one of the challenges about this is that if you have a highly mutated factor that doesn't work very well, keeping it around longer isn't probably going to make a difference. But it's probably no difference for those that you have type 2A von Willebrands that have gotten DDAVP in the past. It's really no different than that. And so this really could fill that space that's been left void by Stimate. In mild hemophiliacs, you're able to raise the levels sometimes in the normal range. Even moderate hemophiliacs were able to get their levels up into the mild range, which may be enough for minor bleeding events like nosebleeds and such. So it does look exciting. Obviously, not everybody responds. This patient didn't respond down here. And the other potential benefit, which I'm not sure they're going to explore, is if you, in a hemophiliac, if you give this drug with a factor VIII product, it extends the half-life 2 to 3-fold. So, obviously that's going to might be a little complex for some patients, but exciting nonetheless. The same company that Benny runs, Hemab, is also creating other products. So not just in Glanzmann's, but also in von Willebrand disease as well. And they've created this monoclonal antibody that specifically binds to this domain down here in the CK domain and doesn't seem to have any hemostatic benefit. And so it shouldn't cause any issues, but it works similar to the BT200 by limiting the clearance of this drug. It's subcutaneous, and this product is, probably going to start phase 1 next year. Don't quote me on that. That's something we're sort of, maybe, it's going to depend on a lot of things, of course. But next year, we should start to hear a little bit more about this. But obviously exciting for the von Willebrand space. There's the fancy animation I put in there. Moving on to hemophilia now is, there's a lot of drugs. I can't cover all of these in the limited amount of time. So we're going to cover some of the ones that are highly likely to make it through. And so let's talk a little bit about the concept. So obviously we've had factor products that give you this nice up and down motion here. Extended half-life products might be able to get you there and decrease the interval. But what's interesting is the non-factor therapies provide this sort of consistent level of hemostasis, although it's not providing you as high levels and not giving you those peaks. And ultimately, gene therapy is going to be what takes over, 20 years from now. That's probably mostly what we're going to talk about at these meetings. It's something that we need to consider, because even though this provides a consistent level of protection, there may be some benefit of having some of these peaks. At least that's sort of what I think. And, these peaks right here, this peak that you get when you immediately infuse, there's probably a long-term benefit. We talk mostly about troughs and attaining these trough levels a certain level, but I think we're going to talk a lot more about this in the upcoming years as we get better products that are able to achieve long times over the near normal range. And so I think that's something just for discussion, and we'll set it up and talk a little bit about it when this presentation's over. But going back to this BIVO1 product here, you can see here again the X10 linkers, which make this a little bit bulkier and make it a little bit less likely to be removed. And then this D' D3 area here, which limits the interaction to VWF. Early studies on this in hemophilia have been very exciting. And this is the series that's completed. So they've completed most of the trials here. They're looking at that study that we just talked about in von Willebrand's. They finished the pivotal trial here. There's an ongoing phase 3 trial in children, and then there's always an extension trial, which I think are probably one of the more important studies, because you often will find some issues that maybe were not seen in the phase 3 trials. So the X10-1 was an open-label phase 3 trial looking in children 12 years and older with severe Hem A. They had to have lots of exposures as PTPs, and they were really just looking at ABR. So really simple study. Is the ABR going to be much lower? And they looked at a lot of other things that we won't have a lot of time to talk about. But you can see here, there are patients that were on demand, and there are patients that were on their prophylactic regimen. And, cutting to the chase, you can see here the median ABRs, and no matter how you looked at it, median or mean, they were actually very, very good. There was a significant reduction, as you can see here. Even when you looked at intra-patient comparison, patients that were reasonably well controlled with factor VIII prophylaxis were able to get an additional benefit with this product, down to about 0.7 ABR, so over 77% reduction in ABR. So it looked very good. And it's not surprising, if you have normal levels for 4 days, you should have significant reduction of bleeding. So that wasn't really surprising. The half-life was about 47 hours. And again, you're spending almost 4 days in the above 40% range. And then you're landing somewhere around 15% after a week. So it looked very, very exciting here. And the phase 3 trial results are probably going to be published next month. And so you'll hear a lot about it. And maybe it's October, but probably September. So let's talk a little bit about other products here. There's almost no data out there other than what's on the investor portfolio. That's one way to look for what's in the pipeline. Companies can't talk about it. They have to talk about it in their investor portfolios. And so there's a second generation emicizumab product that's in development right now. I don't know anything about it. I don't think there's anything out there about it, but something to think about. And then, there's other products that are coming right behind emicizumab. And so Mim8 is a product that's created by Novo Nordisk. Again, it's the same concept of bringing factor 9 and 10 together to form activated 10A. And there's been some optimization and, to ensure that most of the assembly happens on the activated platelet surface. This potentially could limit some of the potential thrombotic events and TMA events that we're seeing with emicizumab. All of this is preclinical data. There's nothing to prove that it's going to be any different. And there's some evidence that there could be enhanced activity over emicizumab. So again, preclinical data, we don't know if that's going to bear out in actual humans. But it is exciting to have more than one product. Personally, I think there should be more than one bispecific antibody. I'm always uncomfortable when there's only one product available because there's been many times in our community where something happened and production issues became an issue. This has happened multiple times with factor VIII products. Currently, the trial is enrolling, looking at hemophilia A with and without inhibitors. And so this looks pretty exciting. It is a different molecule, and patients that have developed anti-drug antibodies to this molecule, potentially to emicizumab, potentially could benefit from this product as well. That's again still conjecture, but at least that's something under consideration as well. And so they optimize both parts of the arms to hopefully limit the interactions when it's floating around in circulation, mostly focusing on interactions when it binds to the activated platelet surface. So that's that product. Let's talk a little bit about fitusiran, really interesting product. This was a presentation that was done at ISTH. And you can see here, there's a sort of a very simple balance that we like to show, and it's much more complicated than this. But, you tip it one way, you bleed, you tip it the other way, you clot. And so, this is something that has been trying to be sort of exploited, is that if you can limit some of the anticoagulants, that maybe you can actually tip it in the right direction. So in hemophilia, you're deficient in these clotting factors are the procoagulants. So it tips to the one direction. And what we're hoping to do is limit some of these or target some of the natural anticoagulants to hopefully improve thrombosis or thrombin generation at least. So this is the Atlas series. And so this has gone through a couple of people's hands previously under Alnylam, now under Sanofi. And so this study has been going on, it feels like quite a long time. It's hard to run these studies during the pandemic. But there have been a number of studies that have been conducted and ongoing, and we don't know exactly when this is going to be approved. It's hopeful that maybe next year we'll see this product actually available commercially, but don't hold me to that because I think we've been saying that for a few years now. One of the things that we learned on this is that the initial target was to really push the antithrombin levels very low. A lot of us actually got pretty nervous about that early on, about that, not knowing what the levels are and pushing it really low. And so what happened was there were some thrombotic events, and there was a bleed in Eastern Europe that was mistaken, or at least it was actually a thrombotic event, and they thought it was a bleed. And so because of that and a couple of other non-fatal thrombotic events, they've actually changed the regimen. And interesting enough, it actually becomes even better, to be honest, because most patients are likely to be treated on every other month dosing, which is about as convenient as it gets. It doesn't get much more convenient other than gene therapy, to be honest. And so, so they changed the goal where they're really trying to target a significant reduction of antithrombin and not a complete obliteration of antithrombin production. And so what this is it's a small interfering RNA. The discovery got these people a Nobel Prize many years ago. And some of the early work was done in picking out flower colors. And so thankfully it's moved on to something a little bit more important than flower color. Nevertheless, so this trial that they had was enrolling males with hemophilia A or B, 12 and older, and they could either be on bypassing or factor prophylaxis entering the study. And so there was a lead-in period here where they're on their standard of care. and then they eventually got fitusiran. Remember, these are patients with either hemophilia A or B. So that's what makes this product a little bit exciting, that it has a complete crossover benefit. And what they saw was significant reductions. No matter how you looked at it, there was a pretty dramatic reduction in ABR for treated bleeds, overall ABR as well. And then if you broke it down into patients A or B with inhibitors, there was a pretty significant reduction. And those without inhibitors, they also benefited tremendously. And so remember, this is a product that potentially could be given once a month or once every other month. It definitely is one of those products that is not going to be for everybody, but I think it definitely has its place. There have been a number of thrombotic events in this summary here. One of them was in a patient that had a stroke. A patient, if I remember correctly, had a history of DVT in the past, probably shouldn't have been enrolled in the study. I don't think anybody knew that except for the patient. And then there was another thrombotic event, an unusual location in the eye that eventually cleared as well. Early on, there was a clot as well in the right atrium of the heart that was potentially deemed to be related to the patient having a central line. The management of that patient definitely was a little bit different than what you would typically see in practice. So I think there was some nuance there as well. But certainly, you know, thrombotic events are what we're looking for because these products have become so good that they're sort of towing the line between hemostasis and thrombosis. And obviously, we want to limit the amount of adverse events. So finally, the last class before we get into the discussion is the anti-TFPI. And this is an interesting class of drugs here. And I think some of the little antibody pictures didn't show up on here. But what we sort of see is it's a sort of a negative regulator here. And you're able to improve thrombin generation by limiting this sort of TFPI that has a negative effect on hemostasis. There are a number of products that have tried to enter the market, one by Pfizer, one by Novo Nordisk, and one by Bayer. And we'll see how many actually make it all the way to the end, to the finish line. Hopefully at least 2 of them are battling for that. Concizumab is pretty far along. There have been a number of trials here. Don't worry about this, but they've been trying to optimize the dosing schedule, which has been a little bit difficult. But if you focus here, when you look at annualized bleed rates, you can see in patients on demand versus concizumab prophylaxis. Again, you're seeing this in Heme A with and without inhibitors and Heme B with and without inhibitors. And so certainly there's a significant reduction in bleeding events if you're measuring all spontaneous or joint bleeds. But again, you know, the study has had some issues with non-fatal thrombotic events. They've had to have mitigation strategies put in place to limit the number of subsequent bleeding. It's not really clear on the website what all those mitigation strategies are, but there certainly have been some. And unfortunately, the only sort of issue or caveat with this drug is that this drug is going to have to be given subcutaneous daily. The initial thought was to try to give it less frequently. But if you give it, you have to give much higher dose, which would put the patient at risk for thrombosis. But I'm sure Novo Nordisk has a lot of innovations with their pens, and so I'm sure it'll be a fairly painless injection, similar to what they use for insulin. And so there's another product, marstacimab here, that's being produced by Pfizer. And you can see here some of the early data here that was shown at ISTH in 2021 from their phase 2 trial. I haven't seen much else out there online, at least. But you can see there's a mix of patients, hemophilia A with inhibitors, without inhibitors, and this one has a pretty good mix here. And you can look at the different dosing regimens here, and you can see here this is Q weekly. And these patients obviously have a lot of bleeding coming into it and seem to have pretty significant reduction of the bleeding events as well. So the big question is going to be whether this translates well into the phase 3 trials. Are we going to see some of the same thrombotic issues that we've seen with the TFPI drugs in this class generally? And not to be outdone, hemophilia B, there's a number of products on the market here. This is standard half-life factor 9. This is Alprolix, where you have the fusion of the FC to the factor 9. This is Adelvion, where you have a cleavable linker that's bound to albumin. And this is Rebinine, in which there's pegylation of the activation peptide here. And they have, some characteristics that they share. Probably one of the defining ones is this is a human cell line versus a hamster cell line. Both Adelvion and the Alprolix probably have a similar recycling mechanism. and pegylation is through a different pathway where it decreases renal filtration. It becomes much larger and bulkier and reduces the clearance of this protein. But there have been some changes that have happened. Revinine recently was approved for prophylaxis in adults and children. I think that just happened recently. And it actually is pretty open. It doesn't say what age, it doesn't say newborns, it just says adults and children with hemophilia B. And so I think most of us will read that as, you know, to be able to offer this to young children as well. I think, you know, as a community, we'll have to talk about pegylation and whether there really hasn't been any data to suggest that pegylation accumulates in children, that it has negative effects, but it's something at least, you know, probably worth discussing. And then Benefix had a label change for prophylaxis in which the dosing is now 100 units per kilo weekly. And so that's something that's available on there. I kind of wish they kept some of the older dosing on there too, because a lot of patients probably would benefit from twice a week. And there's something to think about it, but at least for some patients, this could be pretty beneficial. And to be honest, in developing countries as well, I mean, this is going to be a pretty, I think this should be widely utilized. One, because it's much more easily administered, and there's a significant cost savings compared to the extended half-life products. And then not to be left out, because, you know, this was approved, I think sometime around during the pandemic or right during it. Factor 10 deficiency, we were all pleasantly surprised to see that there was a product available for factor 10 deficiency. And you can see what we had to use before was either plasma, or we had to try to force one of these prothrombin complex concentrates in there. And you can see there are varying amounts of factor products. And generally, we don't like to give you know, products and give you a lot of extra clotting factors that you don't need. That's pretty much what we had to deal with before, and certainly we've all used these products. But it's nice to be able to offer Coagudex, which doesn't have any significant other clotting factors. And right now, as of at least last night when I looked at it, it's available for routine prophylaxis to reduce frequency of bleeding episodes on demand. And it unfortunately just has it for mild and moderate deficiency for surgeries. It hasn't quite gotten the major deficiency, which hopefully that will happen. And, you know, we're hoping that most insurance companies won't be able to read that closely to the in there. So we're going to use it for that purpose anyway. So that's all I have for today. We'll discuss a little bit more at this point. So thank you very much. So I think at this time we're going to have the other members come up, and it'll be open for lots and lots of questions. There's no silly or stupid questions. Ask whatever you feel. This is your meeting, so you should ask, you know, the tough questions.
We also have our online community watching, and so they're going to be submitting questions as well. We'll be happy to run microphones to you all once we open it up for questions. You can also write a question on a piece of paper and pass it to one of the session managers as well. Thanks very much. Yeah. I think Lynn's got some questions too. Yeah.
Well, the audience is warming up this morning. Waking up or warming up. So Robert, I'm interested in the treatment that you were talking about for Glanzmann thrombasthenia using recombinant factor 7. And my understanding is that the half-life is very short of recombinant factor 7. Can you explain a little bit about why giving it 2 or 3 times a week might be beneficial?
Yeah, I think that's a great comment. So, you know, in the study that we did, it was technically a study. Yeah, maybe this one's not as good. Yeah. So in the study that we did, or at least in this series that we did, so it's similar to what you see with the congenital factor 7 deficiency. Certainly the half-life isn't any longer in those patients, but most of those patients benefit from fairly low dose, 15 to 30 micrograms per kilo, 2 to 3 times a week, which significantly mitigates their bleeding. It doesn't make a lot of sense. I think there's probably some long-term benefit once you form the thrombin generation, you get normal thrombin generation. I think that has a long tail to it, and a subsequent benefit that extends past the period of the half-life. So, you know, we certainly were well, we were expecting potentially patients not responding at all to this prophylactic regimen, but we knew some of these patients were getting so many doses after bleeding events that we thought, well, maybe we can actually prevent some of these bleeding events. Maybe we can limit the number of bleeding events. And it seemed to offer benefit for these patients. Certainly a few of these patients. I mean, one of the patients that we had, in Shannon knows him well, when I first got to Emory, this patient was coming in for IV iron every month, was coming in for blood every month just because we knew she was going to get it. So they just went ahead and prepared for it every month. And she was coming in for platelet transfusions. This is, and then she'd had to come in early to get her, to get a type and cross. And then they had to go search for the blood, which took a few days. So this was, this was really tough on her, and she really wasn't able to do much. She was missing a ton of school. And so, once we did this, we put an IUD in her, we put a central line in here, and we tried this strategy, and it really completely changed her life, where she had significant reduction in her bleeding events. She was able to do things. She was able to come off of blood transfusions, IV iron, and platelet transfusions. She still has bleeding events here and there. Every once in a while, she'll have a significant bleeding event, but certainly, she was headed toward bone marrow transplant if this wasn't going to work. And that was going to be tough because she'd gotten so many blood products. So I think it's worked in some of these situations. It's something to think about. And certainly it's something worth exploring as part of a trial. I think we have to try to try these very difficult trials to do, because there's just not a lot of patients. So.
Audience questions? We have one.
If you were to speculate on the second generation Emmy and how it's going to be improved, what do you think we're going to see?
So, Donna, do you mean what we'll see in regards to how, efficacy?
Are they going to be efficacy, safety?
Well, hopefully those would go hand in hand. I think the fear with increasing efficacy is that you're increasing thrombin generation. And so sometimes increased efficacy can sometimes decrease safety. So I think we'll just have to watch those trials really carefully. And I think that it shows the importance too of working with a treatment center that understands how to run clinical trials, so that patients can be watched really, really carefully as we're trying these new agents.
So I think the extension of that idea is even after these products potentially get approved and are on the market, following the effects long term is very important, because we know from factor trials, from emicizumab trials, that we're seeing different things in real world data than we see in the clinical trials. And that holds. I know we didn't talk a lot about gene therapy today, but that holds for gene therapy also. So following patients long term, 10, 15, 20 years after they either start a new therapy, get gene therapy, is going to be really important to make sure that the safety and effectiveness endpoints that we see in the trial actually live up to it in the real life. And Donna from World Federation of Hemophilia and Athens, where I work now, do have a protocol for following gene therapy patients up, commercial gene therapy patients up. And I would advocate that anybody receiving these medications, that enrolling in these registries would be considered standard of care, to make sure that we get this data appropriately.
So I'll ask another question while the audience gears up for their next question. And that is, Robert, you talked about BT 200, and the clinical trial would be enrolling potentially women with hemophilia, women with bleeding. That's a really important health equity issue. So can you talk a little bit about why that strategy was chosen?
Well, and so, we've been trying as a group and a community to try to encourage some of the drug companies to consider, focusing on underserved groups such as women and girls with hemophilia. there's probably a benefit. When you look at mild hemophilia in general, about 1 in 5 mild hemophiliacs in this country are female, and it's probably even closer to 1 in 4. With high dose desmopressin and the uncertainty about things like stimate out there, I feel like this is a perfect opportunity for drugs to come in and target the non-severe population. So I think the community's done a really good job of really tackling issues related to severe bleeding disorders, severe hemophilia. Probably hasn't done as good a job on non-severe hemophilia. And so I think this is a good start to focus on that group. It's an underserved group. It's much more difficult, as you know, to conduct a trial in non-severe hemophilia. Severe hemophilia is really easy. You just count bleeds before, you give the drug, and you count the bleeds after, and likely you're going to have a benefit. It's just not that simple. It's sort of like doing a trial in von Willebrand's. It's very difficult. So you have to look at other things, other sort of outcomes and other measurements. And so we're hoping to look at things like pictorial bleeding chart on heavy menstrual bleeding, other bleeding events, instead of simply counting them, because they're different type of bleeding events. Because you're largely talking about in severe hemophilia, you're really counting muscular and joint bleeds, and you're not, in non-severe hemophilia, you're counting other bleeds. And it's hard to compare nosebleeds and GI bleeds and hematomas. And so that's where it gets a little more challenging. But it makes it exciting because I think that's the perfect space for a drug like this. It could be very useful for that purpose. And you could see it being used for procedures as well. If you give the drug ahead of time the day before, you're likely to have adequate levels, and you can continue to give that drug after. So it's just going to be such a convenient drug if it works out in that situation.
We actually have a couple of questions online too when you're all ready for that. So question 1 is, would there be any benefit for something like HMB-001 to be used in patients with factor 7?
Yeah, I don't think so. I think that's going to be, I think they're probably going to take a look at it. Obviously, it's very early in this, and I think there's some representatives here. You can go ask them as well whether there's some discussion about that. But for factor 7 deficiency, we obviously do have products available, at least one product available, and so it's possible. I think they'd have to really take a look at this potential safety wise. There could be some issues there. But I know they're looking at things like Bernard-Soulier syndrome, Glanzmann's, and factor 7 deficiency. So I don't want to eliminate it as a possibility, but it's something that they're exploring.
Great. I have some more online questions, but I'd like to take one from the audience. Oh, here we go. Hold on. Let me get you the mic. Well, the online community can't hear.
Excellent presentation. I had a question regarding the new product that you mentioned that decreases activity in the anticoagulants in the clotting cascade. What are some of the ancillary, if any, uses of having those anticoagulants in the system that by decreasing we might see consequences from, aside from potential thrombotic events?
So that's a great question, and we don't actually know all of the different things, say, a TFPI does. It has multiple binding domains, and so these drugs will need to be watched very carefully in clinical trials to see if there are off-target effects that we're not anticipating right now. But excellent question.
Yeah, I think, it's a great question because, we're all focused on hemostasis, but some of these things have things that affect outside of hemostasis. And a lot of us get a little worried when you start targeting things like protein S and protein C. What happens when somebody gets really ill? Is there going to be an issue? Can you reverse these when someone's getting sick as well? Because these are important with inflammation, and they're altered significantly when somebody has an infection. And certainly during COVID, we were very concerned that some of these patients could be at high risk. They certainly didn't see any. If you look at the trials, they actually comment on it specifically on fitusiran and concizumab. They didn't see any effect from COVID. And it's not like these patients weren't getting it. They were infected as well. But it certainly gets us a little concerned when you target some of these things, particularly if you get them very reduced, that there may be something that we don't quite understand. So that's why, like Mike was saying before, it's really important that we follow these patients long term. We capture events that happen outside of a 6-month window in a trial, and learn a lot more about this. And it's good that they have all these mitigation plans in place to not severely reduce these things, because we don't want to gain a little bit better improvement in bleeds, but then gain a lot of side effects. So that's a great question.
Okay, I've got one in the virtual chat, and then I'm going to head to the back. So the one in the virtual chat is, yesterday there was a talk on bone disease and hemophilia. What are your thoughts of giving weekly factor VIII for patients on ME? Will the same issue apply to other non-factor therapies?
Also a great question. And I think for those of you who are in that session, low bone density has been seen sort of across the board in hemophilia, but it's not entirely clear why. And so we don't, and in some animal models and some research models, it seems like factor VIII could rescue that bone density to some degree. But we don't know if the effects of non-factor therapies also rescue bone density. So there will be a study starting, I think, very soon from Rebecca Cruz-Jarvis in Seattle that will actually be looking at bone density in patients on emicizumab. So again, the importance of research and participation. We can't know the answers until we do the studies.
So I was lucky enough to participate and help design some of the mice study that looked at bone density in mice that didn't either produce factor VIII or factor IX. And the endocrinologist that we were looking at, that we were working with, said that the teenage mice, the equivalent to teenage mice, right? So they were 20 weeks old, not 20 years old, had bones that were essentially of the same health of postmenopausal mice. So very significant impact. That bone health improved with supplementation with factor VIII. There was no difference in the activity of the mice. So it wasn't that the mice who didn't produce factor were less active because they had bleeding. The mice are so light that they actually don't bleed into their joints spontaneously. We actually worked with Len in his lab back in Chicago to look at some of these effects also. But what I would advocate is don't overgeneralize mice data into human data. There are a lot of ways that mice are great models for what's happening in human being, but there are also a lot of ways that mice models are not the most appropriate models for adults. So the trial that Rebecca is starting, that we're actually pitching to extend through the entire Athens affiliate network, is the way that we're going to have to do this. We're really going to have to look at this in humans.
So as the one person on stage here without an MD, I think I have a basic science question maybe for all of you. And to your point, right? Mice are not humans. And there's the amount that you can generalize from a mouse study or any sort of a preclinical study. You have to sort of take that with a grain of salt. But Robert, I mean, I think you also alluded to some of the challenges of doing clinical studies for some of these ultra rare disorders, right? I mean, there's just not patients to get on study. So I wonder, I think the question is for all 4 of my colleagues here, what are some of the strategies that we can use, whether they're, non-clinical studies or other things, to sort of overcome some of that challenge with trying to do this development and try to get adequate safety signals in ultra rare disorders? Like, how do we solve that rare patient problem?
Yeah, let me comment first on about the bone mineral density stuff too. Is so we there should be a paper coming out. So Divya is a provider, a doctor in Arkansas. We were able to look at some claims data sets on focusing on women hemophilia carriers, and it's always hard to tell whether they have factor deficiency when you're using different codes. But we looked at patients with von Willebrand's as well, and those that are carriers, and there was definitely a worse bone mineral density in that population compared to the others. We're a little surprised to see it in von Willebrand's. I think we have to do further studies. The coding for von Willebrand's is not that great. There's basically one code for all of the types of von Willebrand's, but certainly you saw a lot more procedures. And even in a Swedish study, they saw there was a significant number of orthopedic procedures, joint-related procedures, and bone procedures done in carriers compared to the rest of the female population. So there's probably something there. On the counterpoint, you could say, well, what about hemophilia B? Like, if factor VIII is so important, then why do people with, you see the same issues in bone mineral density and hemophilia B, and they have a normal factor VIII? So I think, a lot of people feel that it might just be thrombin generation, the ultimate goal. It's probably very complex based on physical activity, on how you're feeling, based on what products you're on as well. So again, like Mike said, we can't generalize. But going to your question about doing trials in rare patients, we certainly can't do it the same way that we do a hemophilia PTP study. That is easy to do. A new drug comes on, you just plop it in the formula. You probably have to change about 20 words, and it's basically the same trial. The only decision you make is there a lead in or is there not a lead in. So those are pretty straightforward, but we can't force that model onto non-severe hemophilia, onto rare bleeding disorders. And for these rare studies, I think there has to be something happen. We can't keep doing studies the way we're doing, contracting with 30 institutions. That takes like a year now to do, costs an insane amount of money. And half the sites don't even enroll a patient. But they all charge a lot of money just to get those. And then also the site spends so much time doing it, and then they don't get any benefit out of it. So there has to be something that has to change for this. We have to be able to do some of these studies. And, we're going to have to figure a way. We're going to have to cut out some part of the universities in this, because it's just not sustainable. We can't keep doing trials this way. We can't have a study in a rare disorder and open it at 45 sites to enroll 10 patients. That's just not sustainable. And then it shows up in the high cost of the drug as well. And so something has to change for sure.
So I think the other point that needs to be made is that there's some degree of community participation. So if you think about the data that was generated from COVID trials, there are hundreds of thousands of people who enrolled in COVID trials. If you look at heart failure or cardiac trials, when people get stents, for example, put in, there's 40 or 60,000 people enrolled in those trials. If you look at cancer trials, 20,000 people enrolled in a trial. You look at hemophilia A trials, there's 150 people enrolled. So it's capturing everybody that's available. In rare disease, we as a community need to work harder to capture every available person to get into these trials. And that means reaching out to the entire community, not just those that are currently seen in a hemophilia treatment center, but those that are currently not seen in hemophilia treatment centers. Your data is equally important, and your contribution to rare disease trials is critical. And if we don't have that kind of enrollment throughout the entire community, we need to get close to 100% of people, for example, with factor 13 deficiency enrolled in a clinical trial, not 8 or 10 people, which is clearly not representative and doesn't have that health equity lens either. We do clinical trials basically in white males of, a particular age, because that's who, has had hemophilia. Robert made a great point. We need to have trials of women with hemophilia. We need to have representation of other diverse populations who are impacted by these diseases. So as a community, we need to work harder, but clinicians, the physicians up here, the people who run hemophilia treatment centers, their hands are tied. They can only enroll the people who present to them. You need to participate. So you need to bring your friends, your family members in to participate in these. So this is your call to action. You want these drugs, you need to participate.
And can I make another point? So when we come to you, when our surveillance coordinators come and say, will you sign up? Will you fill out this extra paperwork for us in clinics today? Please say yes. Each person is so unique, whether you have hemophilia or von Willebrand disease or platelet dysfunction or another factor deficiency. Your disease is different than someone else's. You might have a different mutation. You might respond differently to these medications. So unless you participate, we are not going to know how you respond to these therapies.
We've got a lot of questions. I've got one here that I'm going to pass the mic off to. I got a note pass and a bunch in chat. But one of the things I would love it if you all could do before we continue is to just do a brief introduction. I'm so sorry we didn't do that. So everyone knows who you are and how and what your role is in the community, where your treatment center is, et cetera.
All right. I think everybody knows Robert already. I'm Amy Dunn. I'm a pediatric treater at Nationwide Children's Hospital and Ohio State University in Columbus, Ohio.
Len Valentino, I'm the president and CEO at National Hemophilia Foundation. I'm also a pediatric hematologist who took care of children, adolescents, and adults for almost 35 years in Chicago.
I'm Kevin Mills. I'm the Chief Scientific Officer for NHF. And as you heard, I'm the one on the stage here without an MD. So I treated 0 patients, but lots of mice.
And my name is Michael Recht. I am also a pediatric hematologist. I am the Chief Science Officer for an organization called the American Thrombosis and Hemostasis Network, or ATHN. ATHN is the organization that helps the treatment centers across the country collect data, both for the CDC surveillance project, for our federal grant through Health and Human Services that allow us to fund the treatment center. And then we have clinical trials that we have originated through ATHN itself. And I recently moved from the center in Portland, Oregon to the center at Yale University.
Thank you.
So there have been some questions with the Factor IX products and how they function in the extravascular space. What's the latest on that? And if we started to see how it's manifesting in real clinical outcomes with real patients?
Yeah, I think Robert has the strongest opinions.
So I think what he's alluding to is that, a couple of years ago, we noticed that some patients on certain extended half-life products did not get the benefit that they were getting maybe from previous products. And I think, this just goes back to, what we knew about hemophilia B. I think a lot of people just said, oh, it's just one different, it's got a longer half-life. They didn't understand the differences between the inherent differences. And so there seems to be, and again, this is based on lots and lots of mouse studies. There's a one baboon model too, so just to throw that in there. But, there seems to be significant benefit. And when you infuse Factor IX into a human, for sure, we know this, it goes into the extravascular space. And, if you talk to a lot of the doctors, specifically in North Carolina and Davide Matino in Canada, they really feel like that Factor IX is probably the most hemostatically important Factor IX. And so there's some questions with some of the newer products, whether it goes as quickly into that space, whether there's some interference with that, and whether that has a potential negative effect on control of bleeding. And you may see, we saw some discrepancies in which patients had higher levels, but continue to have bleeding events. And so we published this data, and there's additional data published out of North Carolina. And I think it just goes back to like a lesson. I think sometimes these are from drug companies, we get focused on all this sort of shiny objects. Oh, we're going to focus on trough. We're going to focus on levels. We get focused on all this other nonsense. But the ultimate goal is control of bleeding, having low ABRs, and making sure patients feel confident in conducting their, normal activities. And sometimes we get distracted by levels and think, oh, we got great levels. That means they're never going to bleed again. So we have to be very careful about that and focus on control of bleeding. And one of the things I worry about is it affects how people feel about their bleeds, or at least the feeling of joint bleeds and muscle bleeds. Because if you look at some of the trials, they have pretty high arthralgia rates or joint pain rates. And everybody will always say, oh, these are older guys that have target joints and such. But you don't see those on previous trials. So I think that's a really silly argument, to be honest. And I hate when people say that to me immediately. It's like a knee-jerk nonsense reaction. And so I think what's happening is that patients are having some bleeding events that they don't feel the same way as when they were not on that product, and their joint bleed feels differently. And maybe they're not treating it as quickly, or not taking it seriously, thinking maybe it's just joint pain. And we saw this personally at our center, where a kid was having achy joints, and we brought him in. He actually had joint bleeds. And we thought, oh, he shouldn't be having this. His level's like 15 to 20%. So I think it just goes back to anytime you switch a product on there, you should be very careful. There should be close follow-up. The nurses should be calling to evaluate whether they're having these unusual breakthrough bleeding events. But certainly there are those products we're talking about, majority of the patients were doing very well, but there's a small population that didn't seem to respond as well. And, there's a lot of discussion about whether it has to do with their type of mutation or not, whether it interferes with infused factor 9. So that's a whole other discussion. So nothing new.
I'm going to make another plea to you. When you come to clinic, bring your bleeding logs, fill them out. It really does help us. There are online bleeding logs now that you can link directly to your center. So we can't know these breakthrough bleeds are happening unless you keep track and tell us. So please, we really do use that information to try to tailor your therapy to you.
Okay, I've got one in the chat that's been anxiously waiting. We're going to get to everyone. It seems like the soon-to-arrive gene therapies will bring a lot of change to our community. How will you all approach the difficult choice of potentially giving patients the choice to live without hemophilia, but also keeping the lights on at your practice?
Keep the lights on in your practice.
Oh, keep the lights on. Oh, okay. So yeah, that's a loaded question. So I'm gonna start with the first part, which is about choosing patients for gene therapy. I think the gene therapies are exciting. We've been talking about them for decades. They're finally here or right around the corner. But these therapies are not for everyone now. There are still a lot of unanswered questions. I think that there are going to be patients that are good patients for gene therapy. I think that there are patients in whom gene therapy is not right for them yet. So you need to have conversations with your treatment center staff who know you about these drugs. They are not a one and done. There is no guarantee it will work for you. You may have complications. You need to have all of the information before you make a choice like gene therapy. But talk to your treatment centers. I think that's the most important thing. It's not a one-time conversation. It's a series of conversations over time where you have time to take in all the information, go home, synthesize things, come up with other questions, come back to the treatment center. So it's a series of conversations with people who have experience doing gene therapy as well. So coming to meetings like this, extraordinarily important, where you get the chance to interact with people who are doing gene therapy at their centers. So investigate it, talk to people, get as much information as you can before you make your decision. In terms of keeping the lights on, so what that question is alluding to is the way that hemophilia treatment centers are funded in the United States, which is often through the 340B program or selling factor products to their patients. And so it's unclear right now how the benefits of gene therapy drugs will impact hemophilia treatment centers. But you know what? I'd be happy to put myself out of business. That's my goal.
But remember that you're not going to put her out of business if you have gene therapy, because there's only a select population of people with hemophilia A or B. We haven't talked about all the women who are not going to be eligible currently for gene therapy, all the people with von Willebrand disease, all the people with Glanzmann thrombasthenia, all the people with other rare bleeding disorders, and potentially other blood disorders. So there's a lot of work that still has to be done. Plus, if you get gene therapy, as Amy said, it's not you get it and you're gone. You still need follow-up. So there's a lot of work that still has to be done. The lights are not going to go off. Don't worry, we're going to stay around.
I do. So, like Len, I've been treating people with inherited bleeding disorders for decades and have been honored to run treatment centers at a couple different institutions over that time. And I think right now, based on Robert's talk today and then the gene therapy that we really haven't talked about a whole lot in today's session, we're in the most exciting time of in therapy for inherited bleeding disorders since I've been in this field. And part of that excitement not only is the potential availability of new, safe, and effective therapies, but it's also the unknown. And I think that this idea of patients, families, treaters, all being in partnership to really figure out what's going to go on with these new drugs. There's so many questions that we're going to be able to answer. If we do the trials appropriately, and if we have the people who are involved, who have the conditions, their family members, their caregivers, involved in helping us ask those questions. 30 years ago, when I started my research career, I answered the questions I thought were cool. Past 10 years, I've completely changed the way I do research. I'm now involving people who have inherited bleeding disorders from the very beginning of the trials that we're designing. So we can make sure that we're answering the questions that is important and impactful for the community, not just the cool science stuff, but the stuff that really is going to make a difference in the day-to-day life. You know, we have great drugs that if you look at them on average, people don't bleed anymore. That doesn't mean inherited bleeding disorders do not have an impact on people's lives. And, you know, for hemophilia, for example, we're always talking about spontaneous and traumatic joint bleeds. We're not paying attention to nosebleeds and how many times you miss school, or how many times you have to be taken home from school, or your parents missing work because your nose is bleeding. So those are the kind of questions we have to continue answering, along with the safety of the new products.
And how do we know what those questions are? We have to ask you, right? We need to talk to you. One of the ways that we can get those answers from you is something, if you check out the NHF booth, is Community Voices in Research. And this is your opportunity to tell us what's important to you. What's your life experience with a bleeding disorder? How is it impacting your work, your lifestyle, your daily activities, all of the things that are important to you? We don't know. Dr. Dunn has no clue if you don't tell her, right? So Community Voices in Research is a way that you can share your lived experience with the community and get feedback, benchmark yourself against other people that have similar diseases like you as well. So check out the NHF booth. You might get enlightened.
Okay, so I have another question. Do you want to say
Well, quickly, I was just going to make sort of an additional comment to both of those, and I was going to actually talk about the cool science, right? So we talked a lot here today, or we heard a lot about future therapies, things that are coming, but it's really sort of the near future, and there's also a far future. And I think from the nature of some of the questions, and certainly from Robert's talk, there's still more we don't know about coagulation and bleeding than we do know. We sort of think of the coagulation cascade as sort of this linear thing, but there are probably lots of functions completely outside of bleeding entirely that are a complete mystery to us. And Mike said this is one of the coolest times to sort of be treating patients and sort of bringing these innovations to patients, but it's also a amazing time to be doing some of that really basic science and that basic discovery, right? We have the power of genomics. We can sequence whole genomes in an afternoon now, and that gives us the power to, I think, really understand the complexity of these disorders, and actually will really help us to drive new therapies and new innovations in some of the rare disorders too, where our understanding is really poor. But we think about hemophilia and BWD and these bleeding disorders as single gene diseases, but no two people bleed alike, right? And there are 25,000 other genes in your genome that are all interacting to influence how you bleed, the degree to which your factor levels make a difference, how you bleed, why you bleed. And so I think we're also in a really cool time of trying to unpack and understand that basic biology. And it really matters, that biology, because that laboratory work that's happening today is the cures that are coming, and the treatments that are coming tomorrow. So yeah, the science, the basic science is pretty darn cool too.
I totally agree. And this is the idea of how cool the basic science is, can't be understated. The biggest issue I think that we need help from this group here is the majority of this basic funding, basic science funding comes from the federal government, comes from the National Institute of Health. And there are different divisions within the National Institute of Health. The one that we sort of fit best in is called the National Heart, Lung, and Blood Institute. The National Heart, Lung, and Blood Institute funds research into lung disease, cardiac disease, thrombosis, and blood disorders. Guess which one has the smallest number of people? So when we have those 40 or 60,000 people cardiac trials, it's really difficult to convince the NHLBI that, you know, a trial with 100 people with a bleeding disorder is impactful. So, you know, when we talk about Hill Day and advocating through your state and federal government, that's really where we need work and to partner with everybody in here today, because we need help to convince the National Institute of Health that this type of research is important.
So just one quick question to the audience. How many people in this room remember My Life, Our Future, or participated in My Life, Our Future? Even more importantly, show of hands, people that participated in My Life, Our Future. It was like a decade ago, wasn't it? Do you know that only in the last 3 years have we started to see the publications from that project? Some really important research has come out in the last 3 years now from My Life, Our Future. It takes a while to do that, but that's the value of participation. A study that was designed to genotype people for free, give you your factor VIII genotype. But now we're getting important gene sequencing information. Really cool stuff is coming from your contribution to My Life, Our Future. Can't say any more about how important your participation in research is.
Okay.
Take it away, Brendan.
Okay, I'm ready. I'm ready. Okay, so I had a note passed. Can the treatments for ultra-rare platelet disorders eventually be used for more than just Glanzmann's and BSS? Because all of our treatment options are the same.
I think the easy answer is maybe, but we won't know, right? Until we study it. So we need patients with all sorts of different disorders. So we can have the scientist say, well, there's a mechanism that might work for XYZ disorder, but then we need to link the scientists with the patients who actually have the condition, with the treatment centers who can open the trial and follow the patients. And that's the only way we'll know, is working together. But hopefully, yes, we need treatments for everyone that has bleeding, not just people with severe hemophilia, right? We need treatments for everyone.
And how do we know that? By going to your treatment center and enrolling in community counts, right? Because that's how we know how many people with Glanzmann thrombocytopenia, Bernard-Sillier, and all these other platelet disorders there are. We can't make a plea to a drug company to do a trial when there's 4 people in community counts with a particular disorder. We need more people to participate in that, and we need more of those lived experiences through community voices and research.
Well, and then also enroll in Athen 10, which is offering free genotyping for patients with rare disorders. Genotyping is incredibly important, as you heard from Len. Your genotype might be very different from someone else's, which might mean that your treatments might be different than someone else's. And also, as we look toward that far future, I think is what Kevin called it, gene editing is out there. So knowing your genotype is going to be important, because ultimately, if we can fix your individual mutation, that would be the ultimate cure, right? But we got to have the genotype before we can do that.
So these questions are so great, right? Very specific questions that get at massive issues. As the chief science officer of Athen, I don't want to be up here and plug Athen and asking you to do everything. What I really want to say is, if you're going to your treatment center, or you're not seeking care in a treatment center and going to your hematology provider, and you're not being offered to be counted, to participate, you have to ask your provider why that is. Why are you not offering me at least the opportunity to participate in these kind of registries, in these kind of trials, in the kind of surveillance, so we can help move this entire community forward.
This is going back to the factor 9 conversation. Do we have an understanding of why certain individuals don't have a good reaction when it comes to breakthrough bleeding or trauma using long-acting products and have to depend on standard-acting products to take care of those bleeds?
Yeah, I think we don't know exactly. One of the, at least the hypothesis that's been put out there, and this is where, you know, we haven't had a lot of people interested in funding these projects, and certainly, hopefully, there's some other docs out there that are interested in taking this kind of project on. But one of the things that came out of it is whether it's the mutation that you have inherently makes a defective factor 9, and whether that interferes with the infused factor 9. And so there may be a variability in response. So those with like a, you know, maybe a large deletion mutation, they definitely have a lower recovery, meaning the factor 9 goes into those spaces. There may be some polymorphisms in how it binds to collagen type 4 as well that we don't know about. But this is where I think, you know, we need to spend a little bit more time to try to understand this, because certainly the majority of the patients are going to respond to most of these products fairly well. But there's that small portion that just won't respond very well. And it's obviously, we don't want to have patients, you know, having lots of unnecessary bleeding for 3 to 6 months before we can sort it out. And that's why it's really good to communicate with between your facilities. And, you know, we never had the intention of putting somebody on extended half-life product and then having to use a standard half-life. We sort of talked about it. It seemed like way too complicated. I get concerned that people are going to mix them up. But certainly there are patients that seem to respond better that way. And so I think we do have to do that. So again, this is where getting involved in registries, I think there has to be some effort in doing genetic testing. There hasn't been a lot of effort in trying to understand genetic testing. And I'm hoping that there's some other lab-based scientists that are interested in trying to look at these and maybe predict who is going to respond better to one standard half-life product, or maybe respond with no issues to an extended half-life product. And so that's something that we've been talking to. I know I've been talking to Dr. Stafford at UNC, who's had some ideas, but a lot of them are just not easily done in humans. Very easy to do in mice, so not easy to do in humans. So
easier to do in mice, not super easy to do in mice either. But I think that's actually some of the cool science, right? I mean, I think this question exactly highlights the unknown complexity, right? The biological mechanism of how this works. We really don't know. And so I think, one, to Len's point, on everybody's point, we really need data and research participation in order to be able to unpack some of that complexity. But I think also, to Mike's point, we need research funding and the motivation to do that laboratory work. Because I mean, there are only so many patients and only so many genomes that we can genotype, and we can do a lot more in the laboratory. But it's expensive, it's time consuming, and we have to be patient, because the time for that basic research to turn into tangible clinical applications is a long time. But I think it's we need to hear from the community about what's important, what matters to you, and then it's a community effort. We need to do the clinical science and the basic science, and we need to be able to connect those things together.
Let me close that loop real quick. So you might think, what can I do? Well, you could give a big donation, of course, to fund research, but you could actually do something that's more important. You could participate in Washington Days, right? That's where your voice gets heard. That's where we make a plea to our congressionally, elected representatives. Give some more funding to NHLBI. Participate. Have your voice heard.
Love it. Okay, so we've got a question in chat. Hold on, another one popped in. Thrombotic events have always plagued the anti-TIFP drugs in the trials. Were there any risk factors identified in any of these trials? And why the predisposition to arterial events more than venous events? Any speculations on this? Thanks, Robert.
I think I think the simple answer to the question is what we've all said. We don't know. So if you look at all of the drugs that have been developed to inhibit tissue factor pathway, most of them have failed, and they failed because of blood clots, because we don't really understand what we're doing, right? That's part of the problem. We need more of a better understanding of basic science. You know, tissue factor plays a critical role in blood vessel development in early embryonic life. If you interrupted tissue factor in an embryo, you would not have blood vessels. You wouldn't develop a heart. But somehow that gets turned off. The importance of that gets mitigated as the embryo develops and then later in life. But we don't really know. Is that completely turned off? Maybe there's some risk for thrombosis there that we don't really understand. We need more research. Robert could answer the question from a clinical perspective, I'm sure, but let's take another question.
Hi there. So my question is with respect to gene therapy. I know that it's a really exciting new treatment option for some patients. My question to you as providers is, how are you going to approach gene therapy, specifically some of the options that require the long-term steroid use? Having us just talked about patients being at high risk for this, for their bone health and whatnot. So how is that long-term steroid use going to affect it, and how are you going to have that discussion with your patients?
So this is where these conversations with your provider are so important, because not only do you need to know what we know about gene therapy and its potential benefits or risks, but you also need to understand what we know and don't know about the potential use of immunosuppression, mainly with steroids. Some trials have used some non-steroid drugs as well, and steroids are not without complication. And so low bone mineral density is one that's very important in people who are on long-term steroids. So you need to talk to your providers. You need to have a sense of what your underlying bone health is, I think, before you would want to consider long-term therapy with steroids, which is likely to be needed in most of the gene therapy trials.
I'd like to reiterate a little bit of what Amy talked about before, and the time and the true assessment of understanding of what it means to receive a commercial gene therapy product. You know, I think this might be one of the few times where consenting someone for a commercial gene therapy might be actually more difficult than for the clinical trials, right? The clinical trials, we can go in there and say, you know, spend all the time and make sure that we think that a participant understands what's going on. But in the end, it's like, oh, we're in this together. We don't know exactly what's going to happen. Commercially, that's very different. And if and when these vectors are approved, they're going to be approved with lots of questions still unanswered. But spending millions of dollars for a drug and not exactly knowing if you're going to respond to it and not knowing those long-term effects is really going to change the way I think about approaching patients when they express some sort of interest or when I talk to them about gene therapy options in the future. So again, going back to what I said about this being a really exciting time, that to me is really wonderful, right? The more time I get to spend with my patients, the more time that we can really explore what someone's goals are, and whether gene therapy would meet those goals. That's all time well spent.
Well, Mike, just as a follow on to that, maybe comment on how long it's going to be until we have the answers to some of those unknowns that you just talked about.
Yeah, I think the question of the immunosuppression, that should be answered fairly quickly, right? Being on steroids, whether for a couple of weeks or a couple of months or a year, we sort of know what the implication of that is, what it means in the middle of a pandemic, what it means for someone who might come in with not the healthiest liver or not the healthiest bone. That's the kind of stuff we need to understand. The real issue, I think, is ensuring that anybody that receives a current version of gene therapy vector or any gene therapy vector in the future is followed for a prolonged period of time. And is that 10 years or 15 years or 20 years? I don't know, but it's a long time. And I think one of the risks that we run are people are going to get their gene therapy. They're going to have their immunosuppression, whether they need it, if they need it or not. They're going to end up with whatever factor level they get. They're not going to bleed as much, right? If they respond, they're not going to bleed as much. The risk of losing people to follow up, I think, is a big risk.
Okay. We've got one last question. Our time is over. And I'm going to pass the mic to one of our NYLI participants, Tyler.
So I have a couple questions about BT200. So you guys mentioned that it's more commonly, the trials are more commonly done with female patients with mild hemophilia. Is there an equal amount of male patients in these trials? And then my second question would be, what phase is this trial in?
So the trial, the trial originally were done in males with hemophilia and von Willebrand's, and they had, obviously, the early trials were done in normals patients just to demonstrate that it works. And so one of the things that we were talking about, just to be clear, is that we wanted to shift some of the thinking that the pivotal trial can be done in women. It gets more complicated. We were talking about this before. You have to do these reproductive toxicity studies. Those are extremely expensive. They're often done in these non-human primates, and these can be millions and millions of dollars to do these studies. Because, and the other part of this is that women, they often have to have barrier control, so they have to be on some form of birth control, which could also convey a potential thrombotic risk. So it gets complicated in having women in trials. It's not as just simple as like, oh, we need to have women on trials. We never do. It gets more difficult to do that, and there has to be a lot of preparation for that. But we're at least trying to get companies to prepare for that, to consider that early and consider that this is a good group to continue to study this in. And so the trials that are going to be upcoming are going to be phase 3 trials. So they're actually going to look for efficacy. They're going to look to get a label for this and eventually, hopefully, get approval from the FDA if it is successful. So stay tuned.
For those of you who ask a couple more questions in chat, we will do our best to get some answers put out there for you. Thank you all for being here today. I thought this was a great session. I hope you enjoyed it as much as I did. And I got a whole lot of steps in today. That was really good. I always say we have one of the smartest communities in the world, and you did not disappoint today. Your feedback matters to us and helps us plan events like this and sessions like this. So please tell us what you think. If you could go out on the app and fill out your survey, complete the evaluation, and you'll be entered into a raffle for a $75 gift card. We had 3 winners yesterday, and raffle winners are selected each day and will be emailed their prize. Thanks again, everyone, for your participation.